It looks like it's going well. Please confirm when you have it.
We got the message of live, but still not.
Yeah, it's working through it in my screen. Now it looks like we are live, correct?
Not. I'll refresh here. Give me a sec.
Yeah, we're live. I seen it myself. So somebody else confirm, are we live?
I don't see it, Eduardo. Just give me a sec. I'll try a different one here.
I think we are. I see it.
Yes, it shows up on my computer. Yeah.
Right. Thank you. Thank you.
It does.
So with that, like we always do, I would like to quickly mention what we have accomplished last week. Last week we had Josh Firth from the University of Oxford presenting on the behavioral ecology of contagions, and it was a wonderful talk, very well attended, and we had a wonderful discussion. As you plan to join us next week, October 10th, we will be hearing from Aubrey Kelly from Emory University on neural mechanisms underlying grouping behavior in spiny mice. Today, we have David Queller from the Department of Biology at Washington University in St. Louis, who will be talking about cheating in a social microbe. And as we always do, I want to share a little bit of what David has been doing all these years. We were talking, we met about an hour ago, and it's quite remarkable, quite exciting to see the very eclectic training and exposure to all kinds of intellectual environments he's had, starting with a BA in the history and philosophy of science. So I think that looking at history understands so much of what he's done since then. So he started with history and philosophy of sciences, then he went to Michigan for his PhD in botany. And spent after that 10 years at Rice University in Texas. And from Rice University, he moved to Washington University in St. Louis, Missouri. I always try to find something that makes our speaker different. You will remember him because of that, maybe. And one of the things that I noticed, browsing through his extensive CV, is the list of his PhD advisors. And I'm sure that at least those of my age will just have your jaws drop. I mean, his advisors were Richard Alexander, W.D. Hamilton, and Warren Wagner. So no wonder how and why he got such amazing training and background on the topics that will be center to his talk today. What is quite remarkable as well is that he mixes and he goes back and forth all the time between very, very thorough empirical research and theoretical research. And you really just have a quick look at his extensive list of publications. I think that I was looking at a CV from 2018, and it's well over 200 publications. And there you see all kinds of papers with a focus, again, that he will discuss today on social evolution, on kin selection, altruism, reciprocity, parent offspring conflict, major topics in the history, actually, in the history of behavioral ecology with attention to the evolution of social behavior. He is a fellow of the Animal Behavior Society, the three societies I'll mention in the US. He's a fellow of the Animal Behavior Society, the American Academy of Arts and Sciences, and the American Association for the Advancement of Science. Something else that's quite unique is the funding support he's got over the years. I counted 19 NSF proposals funded and completed. Today, like I said, he will present to us the work that he studied about 20 years ago, because he has had assistants, social insects, and he continues, of course, thinking and working with them. But about 20 years ago, he decided that he needed to try something new, as he was telling me. And so he started working with amoebas. And that's, I understand, will be the focus of his talk today. So with that, David, thank you so much for joining us at FINE. We all look forward to your presentation, and I invite you to take it from here.
Okay, thank you. Let's try to get this running. Are we seeing the right thing?
Not yet. Now we are.
Okay, great. Okay, today I want to talk to you about the topic you see up there of cheating in a social microbe. And I want to start out by acknowledging that most of this, well, all of this work is collaborative in some sense, and most of it is collaborative with Joan Strassman, who actually gave a talk in this series a couple of years ago. I've striven to not overlap entirely with what she talked about before, but it will overlap some. But I'm going to try to develop some themes that kind of run throughout the talk. Included is how the field has developed, how we started, and so forth. The theme of microbes versus macrobes. That's a word you may not have ever heard before. It's a word that microbiologists use to describe the things that you guys study. A little bit about genetic and cell biology tools that we can deploy here. And then some thinking that will go through the entire talk about work in the lab versus work in nature. I'm going to start, we used to work on social insects, right? And we switched to social amoebas. And the work on social amoebas goes back beyond this guy, John Tyler Bonner, but he was important in many ways. He was the main social amoeba guy before they were even called social amoebas. He was at Princeton. And I think almost everybody who worked on social insects, or at least on the question of altruism, was familiar with cellular slime molds or social amoebas from John Tyler Bonner. And the reason wasn't because we all wanted to read his papers on chemotaxis or something like that, the stuff that he did his day-to-day work on, but it was because he's such a broad thinker and he wrote books about culture in animals and development, lots of topics, all of which would include aspects of his work on social amoebas. So we knew about these things, and they were out there. We'd always thought they would be interesting to study. But why did we switch from working on social insects to working on social amoebas and being totally clueless novices on that? Bunch of reasons, and I'll review them very quickly. We were kind of in search of a new direction. I won't say our ideas were running out on social insects, but felt a little bit like we were doing more of the same thing on different species and so forth. We had a grad student who wanted a lab project. He wasn't trained in the field and didn't want to do that kind of stuff. We realized that a Dictyostelium genome project was already underway. This was back in 2000, before there were many genome projects. So there was the prospect of having a finished genome to work with. There was a friendly Dictyostelium listserv where you could post questions, and Joan was particularly good about doing that, and people would answer our crazy questions. And then it turned out that somebody, finding out what our interests were, offered, a guy named Dennis Welker offered us a bunch of wild clones. And I should mention that Dictyostelium is a model system for cell biology and development. A lot of labs work on it around the world, but they all pretty much work on a single lab clone. We were interested in what goes on in nature, and this guy offered us a bunch of wild clones, and that was an opportunity we didn't feel we could pass up. And of course, the main reason, I think, is that we thought there were exciting questions about cooperation and cheating, the same kinds of questions we were interested in with respect to social insects, but now in an entirely different kind of organism, somewhere where we could apply, for example, kin selection theory too, where it was totally unfamiliar and would provide very interesting tests. So what are these social amoebas? They are eukaryotes. They're not bacteria. They're not prokaryotes. And in fact, they're quite closely related to the organisms that you work on, closely related on the grand scheme of things. The red arrow there points to the amoebazoa, which includes Dictyostelium. And although the eukaryote phylogeny has changed a lot over the years, one of the very stable things is that the amoebazoans are closely related, a sister group to the opisthecons, which includes animals and fungi. So yeah, they're unfamiliar, but they're not totally weird. What are they? Well, they do, normally they are standard amoebas, and they do that amoeba thing that you're used to thinking about. They divide into two, and then they divide into four, and so forth. So you can develop from these successive divisions clones of amoebal cells. And everything that we do in the lab involves these clones. They do have a sexual cycle. They can go through sex in the wild. We can't really get them to do it in the lab. but that's okay. There's a great deal we can do by having a different clones and mixing them and so forth. Now, that's the vegetative, the so-called vegetative part of their life cycle. Looks like this is going to take a second to load. Oops. Backwards. When the amoebas run out of food, they start to aggregate. Something wrong with this video, I may have to pass it by, but these are a bunch of amoebas, and you can see a pipetman tip there, which is releasing cyclic AMP. And that's artificially releasing the signal that the amoebas themselves use to attract each other and come together in a mound that's going to form a multicellular individual. So they both produce cyclic AMP and release it and sense it and follow it. Here's a backed up version of that same thing with a whole lot more cells that are streaming together into a very large aggregation. So you may have 10,000 or 100,000 cells in an aggregation like this, all streaming together to form this mound. And once they form the mound, it may elongate and produce this slug, which can migrate across the forest soil, or probably more importantly, it can migrate up through the soil to get to the surface. And the purpose of that is to get to a location suitable for producing a fruiting body for dispersal. The area has run out of food, they want to get out of there, and so they're going to produce a fruiting body. And here's a fruiting body being formed in the middle, well, a bunch of them, but you see one there in the middle, shooting up like that. Let me stop it when it gets up to the top. So the fruiting body here consists of a long stalk and a ball of spores at the top. And so of those cells in the mound, roughly 20% of them form the stalk. And in order to do that, they have to die. And so that's the interesting thing about the social thing about these social amoebas is that 20% of this little group give up their lives in order to provide a platform, a tower from which these other cells can be dispersed. And they're dispersed, we think they can be dispersed by water and so forth, but we think primarily by passing insects. The spores they'll adhere to the insects. So here's the overall life cycle starting in the upper right with the amoebas. And remember, the amoebas can divide, so there's a vegetative cycle there. But when they starve, they aggregate together, they produce a mound. The mound may or may not produce a slug, depending upon whether it's already in a good place or not. And then that produces a fruiting body. And the additional feature that this diagram shows is what's really interesting about these from a social point of view. Yes, 20% of the cells die to help the other ones. That's not really unusual in multicellular organisms, right? Most of our cells die in order to help our gametes reproduce. But the difference here is that all of our cells are genetically identical. They're derived from a single zygote. And so kin selection works naturally among our cells very easily. Whereas here in the social amoebas, you've got cells coming together from a dispersed locations in order to form this fruiting body. And so they may consist of different genotypes. And if they do, then there's the opportunity for cheating, which is shown on this slide by coloring the two genotypes, blue and yellow. And in the end here, we've shown an extreme case where the blue genotype has become spores and the yellow genotype has become stalk. There's going to be this evolutionary incentive to cheat and make the other guys produce the stalk and die, whereas you produce the spores and reproduce and live on. So that's the problem that kind of attracted us to it. Now, there were the sort of start of the field, there's a little bit of work before this, but the main start of this field as a social thing with two papers in the year 2000, one of them not by us, but by Rich Kessen's group. And they found, what they did was a genetic screen to look for cheater mutants, and they found one which we call an obligate cheater. Obligate in the sense that it could only survive by cheating. When you put it in a mixture, it will form the spores. So here's a picture where this mutant is labeled in black, and the wild type is labeled in white. You can barely see the stalks because they're all white. The mutant has induced the wild type to produce stalk, and it produces the spores. But the trick is they do better in mixtures, but when they're on their own, they can't fruit at all. That's shown in the picture on the right here. So they're an obligate cheater. Really interesting. Incidentally, throughout this talk, I'm going to mention work from other labs as well. Sometimes when I put a reference from another lab, I'm going to put it in parentheses, so you'll be able to identify that. If it's not in parentheses, our lab was involved in it. So that was one of the papers. The other paper was our own, our first work on Dictyostelium, and it was led by Yang Zhu, a graduate student at the time. Our first project was to see if indeed these things did mix. If they didn't mix, if they were always clonal, then they were like other multicellular organisms and perhaps not that interesting from the standpoint of social competition. But we were able to easily design microsatellite primers to amplify microsatellite loci because parts of the genome were already available. We didn't have to do any extra work there. And then what we did was to genotype both pure clones and mixtures. And we genotyped them at the slug stage. The reason we did that is that in the stalk, the DNA degenerates, and you want to see what's going on there as well. So we're going to look at the slug stage in this point. And so here's what the results look like. If you mix two clones, NC28.1 and NC28.2, 28.1 has this allele, 28.2 has this allele, and when you mix them together, 5 different slugs here, you see both alleles. They do actually mix together. Here's the same for a different set of microsatellite markers. So interesting, there's the potential for cheating there. Next question is, do they actually cheat? And we could apply the same methods, the genotyping method, but in a quantitative way by partitioning the slug, because it's known that the anterior part of the slug forms the stalk and the posterior part of the slug forms the spores. So we could cut slugs and genotype the separate parts of them to see which genotypes were present in which parts. We did those for 12 different mixtures. Here's a summary of the results. To walk you through that, each of these set of bars is one mixture between 2 different clones, clone 1 and clone 2, different ones. And the clones are represented by different colors. Clone 1 is always represented by blue, clone 2 always by orange. The top bar in each of these segments is the representation of the 2 clones in the spores, and the bottom bar is the representation in the stalk. So this structure out here to the left shows the kind of neutral prediction. If they weren't competing at all, and if they were equally good at doing both things, each one would be present 50% in the spores in the top and in the stalk in the bottom. What you can see is by looking at these things is that that's not generally what happened, that typically one of those clones in any given mixture would produce more of the spores would contribute more to the spores than it did to the stalk. And that was significantly true in 6 of the 12 pairings that we looked at. So what I want to pursue now, what that set up was a whole bunch of work where you could do lab work exploring how cheating works in these things, and you could do experimental evolution and so forth to see the role of relatedness. But I'm not going to talk about that today. I want to talk about cheating itself. And interesting work, and we thought this was interesting work, generates follow-up work. And it did this in other labs. And some of this follow-up work was questioning the extent to which we were really seeing cheating or cheating in the same sense that we thought it was. And there were 3 qualifications that different groups suggested about this. One was that it's possible that the 2 clones behave differently on their own. That is, one clone would make more spores on its own than the other clone, and when you mix them together, it would naturally just continue to make more spores without really cheating in a sense, although you could call it cheating. Another one followed a discovery by a different lab that there is a class of cells that are now being called loners, ones which fail to get into the aggregation, and they can survive at least for a short time. They're starving, but they can survive for a short time, and maybe if more resources come along, they can reproduce. And perhaps one clone is making more loners at the expense of spores, and so it's not really suffering a cost, perhaps, from producing fewer spores. And a similar explanation, but with a different trade-off, suggested that one clone might make more spores, but smaller ones were less viable. So you might look like you're cheating and you're getting more of the spores, but maybe your spores are no good. So let's look at each of those three in just slightly more detail. They have not been totally explored yet, but the first one can be ruled out because you can look at the two clones in a given mixture. You can look at the two clones separately, see how many spores they produce, and make a prediction from what they should, what a mixture should be from that if they were doing the same thing. And that's what's shown here in work by Neil Buddery in Chris Thompson's lab. Neil Buddery subsequently worked on in our lab, but this is work from an earlier stage of his career. Did a bunch of mixes. I'm just showing you one of them here, where they did the mixtures at different proportions, including 0% of one clone and 100% of the other clone. And from these 0 and 1 predictions, you could, and counts of the total spore numbers that they make, you could make a prediction of what they should produce if they contributed to mixtures exactly what they were contributing when they were on their own. And here it's slightly different from the diagonal line that would be the prediction if they were producing the same number of spores on their own. They produce slightly different numbers of spores. That changed the prediction a bit. But the actual result is far from that. They're cheating in a much greater degree than this would suggest. So that number one, we can kind of rule out for at least many mixtures. The other ones, it's a little bit less easy to rule out. Let's take a look at that. So this was a suggestion from a paper by Tarnita in 2015 about the loner cells. And what she suggested is that clones may differ in their propensity to produce loners. So there may be some, like this blue clone at the top, that produce very few loners and therefore produce larger fruiting bodies, and other clones which produce a lot of loners and therefore small fruiting bodies. And therefore, when they mix together, you get a lot of red loners and few blue loners, and a lot of blue cells in the fruiting bodies and few red cells in the fruiting bodies. So again, it looks like cheating. Maybe you could call it cheating, but it's not necessarily getting you a fitness gain because you're giving up loners that might be giving you a fitness gain in other ways. The third possible explanation for cheating is that clones may differ in their numbers versus size of spores. You could, if you had a mixture of, again, a red and a blue clone where the blue is producing more spores, maybe that's because the red clone produces fewer larger spores that have higher viability, and the blue clone produces lots more spores, so it looks like a cheater, but those smaller spores have lower viability. This is from Jason Wolf's lab. There is indeed some evidence for that. They looked at the trade-off between spore size and spore number shown in the top in figure A there, and there is a negative relationship, as you might expect. And in figure D, it shows that this relationship matters, that spore number that you produce alone matters for the number that you produce in mixtures. So some of the cheating differences we're seeing in mixtures might simply be due to this allocation differences between clones, between size and number. The right-hand part of this figure, I'm not going to explain in detail, but they measured the viability of large and small spores, at least in the lab, and they produced a model from that examined net fitness based on spore size and spore number. And you can see it looks rather flat. You can produce more spores, but that doesn't increase your fitness because you're producing smaller spores and they have lower viability. You can produce more spores, but that doesn't increase you. Sorry, I said the first part wrong. You're producing, there's no trade-off in the spore size, but the spore number also, if you can produce more spores, they're going to be smaller and they have lower viability, and your fitness is about the same. So there's a potential trade-off there. So the take-home from these two things, we ruled out one of them, the take-off from these two things is that cheating may trade off against other fitness components. And this isn't a surprising thing. This is true for any kind of fitness component in nature, that it may trade off against other things. So in nature, if a cheating mutation had no fitness trade-offs, it should spread to fixation, and we wouldn't see it as a variant. Everybody would have it, and we wouldn't see it as a variant. That raises the question then, and it's sort of a more important question to me, have these fixations been common in nature? Is cheating important in nature, even though the variants that we see in the lab could often be involved in these fitness trade-offs. So the rest of the talk, I'm going to want to spend talking about whether cheating is an adaptation in the wild. And I'm going to use 4 kinds of evidence, and they're going to be indirect kinds of evidence because we can't do the kinds of things that you guys do when you go out and watch your mice in the wild or watch your monkeys in the wild and see if they're beating on each other, or stealing each other's food or something like that. We can't really watch these things in the wild. So that's the disadvantage of microbial systems that you have to pay for when you get all the advantages of the amazing things you can do with them in the lab. So we're going to look at 4 kinds of indirect messages. Is there population structure in the wild which is suitable for cheating? Do clones mix? We're going to look at loss of cheating when selection is removed. I'll explain that in more detail. We're going to look at molecular signatures that you would expect if there is cheating. That is molecular evolution signatures. And we're going to look to see if there are traits that only make sense or primarily make sense as adaptations to cheating. So these are numbered 1 through 4, and I'm going to start with number 1. Is there a population structure suitable for cheating? And by that, we really mean do clones mix in the wild? And for that, we need to define natural fruiting bodies. We didn't know if we could do that. These have always been collected in the field by going out, collecting soil sample, plating it out, and hoping that a single cell or a single spore grows up into a group that you can see. So we went out to see if we could find slugs or fruiting bodies in the field. Here's a picture on the left. That's a younger me on the right, Angela Fortunato on the left, our graduate student. And in the middle is Rich Kesson, who was the director of the lab who did that other study in 2000. He's a biochemist. But when he found out we were going to go in the field and look for these things, he said, I've got to come along and see this. And he had a fun time. Here he is looking at the very first fruiting body that we found. We reasoned that we should be looking first at deer scat, deer feces, and that's where we found them. And here's a picture on the right of fruiting body on deer feces. So then once you find these, you can collect them. You could collect the spores from a single fruiting body and do some microsatellite genotyping on them to see if they belong to single clones or not. So this is just a summary slide showing that an interesting result. About 3/4 of the single fruiting bodies are clonal as far as we could tell. At least if there wasn't a second genotype in there, it was so rare we weren't detecting it. So they're like multicellular organisms of other types in that respect. But another 1/4 of them were chimeric. They had mixtures of 2 or more clones. On average, relatedness was up around 0.9, very high. So kin selection could work effectively in this, but there is the wiggle room for cheating. You do sometimes find yourselves with a different clone and an opportunity to cheat or the opportunity to be cheated. Second kind of indirect measure, and this is one that we did just recently. A bunch of the work I'm talking about today goes back some years. This is the newest work that I'm talking about today. The idea is loss of cheating when selection for it is removed. All of the cheating occurs at the fruiting body stage. If it occurs, nature occurs at the fruiting body stage. So we reason what happens if we remove that stage during evolution. And the general principle is organisms tend to use it or lose it. If you remove a selective force, if you put a bunch of fish in the dark for many, many generations, they're going to tend to lose adaptations to the dark. These cavefish lose their eyes, for example. So we'd like to do that same thing to our social amoebas. And so how do we put them in the dark? Well, what we can do is remove the social cycle. Remember, they also have this vegetative cycle. If you keep feeding them, they keep producing amoebas and they don't fruit. So what we can do in the lab is keep feeding them all the time. Don't let them fruit. The social cycle will not occur and adaptations to it should diminish. They're not going to go away in the short time of an experimental evolution experiment, but they may diminish is the prediction. And so here's the experiment that was done by Tyler Larson, graduate student in our lab. What you do is you start with an ancestral discoideum clone, you plate it out, you let them grow for a while, and then before they starve, you take them and put them on another plate. Again, before they starve, you take them and put them on another plate. And you do this for, I think he did 30 transfers to fresh food plates. And we replicated this, of course. We had 10 ancestral clones and 3 replicates of each. So a whole bunch of generations without the fruiting stage. And we reasoned that the fruiting stage might therefore decline. But the nice feature about this experiment is we had a couple kind of intrinsic controls where we thought we knew what the answer would be. So we could also measure vegetative cell growth. That's something that is actively selected during this lab experiment. The faster you grow, the more cells you get to the next generation. And we think it's also selected in nature. So in this experiment, cell growth should increase, sort of a positive control. Another control is for slug migration. Slugs migrate a certain distance. The farther you can migrate, we really think that's probably a fitness component because you may be deep in the soil and you may need to be able to migrate to the soil surface. And the farther you can migrate, the better. So we strongly believe it's selected in nature, but now we've created circumstances where it's not selected in the lab because they don't go through the social stage in our experiment. So it should decline. Here's the prediction. And then we did it for cheating, where we didn't really know for sure whether it should decline or not because we weren't sure if it was an adaptation in nature. Hence the question mark there. So here were the results. First, for the controls, cell number or cell division did increase as expected because it was selected for in our experiment. Cell migration did decrease in our experiment. So that confirms our notion. It's a proof of concept that things which are fitness components in nature in this experiment should decline when we remove that part of the social stage. And so what happened with cheating ability? We measured cheating ability both before and after the evolution. And we did it in a slightly different way than the microsatellite way we did before. This is the more common method that people use today. You label one of the clones with GFP or a cell tracker dye. You mix them together. So some of the cells will be labeled and some not. And then you examine the spores and count the labeled ones. And it did turn out that Dictyostelium became worse cheaters after going through experimental evolution when they couldn't fruit, suggesting we think that cheating is a component of fitness in nature. The decline wasn't huge, mind you. It was significant, but it wasn't huge. So maybe you could argue it's a fitness component, but not a huge fitness component in nature. It didn't decline, for example, as much as slug migration did. I can, I think I have time for this, say a little bit more about possible causes of trait loss. When you remove a selective force, there are two things that can happen that would cause trait loss. One is just a pure accumulation of deleterious mutations by mutation and drift. And the other one is pleiotropy, that the experiment is selecting for some trait which is negatively correlated with the fitness component that you're interested in. So maybe selecting for faster replication rate leads to, through pleiotropy, a decline in cheating or the other social traits. These are the same kinds of explanations that you see in senescence theories. For example, you've got mutation accumulation and you've got antagonistic pleiotropy. And we actually had done an experiment many years ago that tested for one of these. And so I'm going to back up and tell you about that. It's testing the mutation, the pure mutation part of it. We did a mutation accumulation experiment, which is a semi-standard kind of experiment that people do on usually rapidly reproducing organisms because it's an experimental evolution thing. And the idea is to eliminate natural selection, virtually eliminate natural selection by a random selection of individuals or colonies to pass on to the next generation. So that little green dot here represents a Dictyostelium colony that we're starting with, and you plate it out sparsely. So each cell gives rise to a new colony. So each of those is a little clone of new colonies. You pick one of those at random, plate it out sparsely, pick one of those at random, plate it out sparsely, and so on. And so each time you're picking a clone at random, so there's no selection going on, and only drift is going on because you're picking them at random. And we did that over 70 transfers for a long time, and a total of over 1,000 generations, and I think 90 replicate lines. So a lot of work here. This was spearheaded by Sarah Fox. And what we saw at the end, again, we tested various fitness traits in the ancestor, shown on the left here, and in the evolved lines under the mutation accumulation experiment on the right. I won't talk about the middle ones here. This is what happened to growth in liquid, the trait that was probably being selected. Growth rates were being selected. Growth rates were high in the ancestor and declined in mutation accumulation line. So you remove selection and the trait declines. It's what you expect of fitness components. And then we did it for cheating as well. We tested cheating of the ancestor and cheating in the mutation accumulation lines. Doesn't look like a huge difference because we've had to plot it out on a large space that included an outlier here, but there was a significant difference, a significant decline in the ability to cheat in the ancestors. So we think part of the response, that at least part of the response that we saw in Tyler's experiment had to do with simple accumulation and drift, but I'll bet part of the response was also due to the pleiotropy factor, but we haven't tested that independently. Okay, so that's our second indirect method for testing how important cheating is in the wild. The third one is to look for molecular evolution signatures of cheating. And this diagram kind of plots out some expectations for how selection works. The bottom one, the part C, is the one I want to show you first. If there's no strong selection, no strong directional selection going on at a given time, what happens is what's plotted is genotype frequency on the y-axis and time on the x-axis, so that you start off with an all gray genotype. After some time, a blue mutation pops up. It's deleterious, so it gets selected against. A red mutation pops up, it's deleterious, it gets selected against. Green mutation and so forth. Mutations keep popping up, but most selection, most of the time, is purifying selection and they get eliminated. Of course, some selection may be positive selection that really does something. And there are two possibilities here. And both of these are predicted to be potentially very strong in conflict situations. So one possibility is you get directional selection where a mutation arises. In part A here, a blue mutation arises and it goes to fixation because it's good. And you expect a lot of those mutations in and selection events in situations where you've got conflict, because one party will fix a mutation that allows it to do better, then the other party will fix a mutation that allows it to do better. So you might have a cheating mutation in blue, a resistance mutation in green, and another cheating mutation in red, and a continual kind of arms race. Or alternatively, or in addition, under conflict, you may get a stalemate. You may get a form of balancing selection, or sometimes it's called red queen dynamics, where a cheater is beneficial at low frequencies, but as it becomes common, it's selected against. And so you may get cycles of cheaters and a cheater mutation versus the normal mutation, or you could get just a uniform blue line across this, a stable equilibrium frequency. And those are also expected more often under conflict. So what did we see and how did we do this? We have to find sets of potential conflict genes to test them for enhanced signatures of these either directional selection or balancing selection. And the first way we did this was to do a Remy selection. And this is actually the same procedure that Rich Chesson used in that 2000 study that I talked about earlier to find his obligate cheater. We used it to find what we call facultative cheaters, things that can do okay on their own, but they cheat when they're in mixtures. And I won't explain the details of how we got the facultative cheaters, but the Remy selection procedure is a sort of knockout selection procedure where you have a plasmid that has some antibiotic selection markers on it, and it's got some restriction sites in it. You linearize the plasmid, the DSR shown down here, and you treat the Dictyostelium with some restriction enzymes that open up certain sites and occasionally allow that plasmid to get in there. And when it does, it usually disrupts whatever's going on in there. If it inserts in a gene, it's going to make the gene not work very well. So you can do this for a whole population and get a whole population of many, many different mutants at different sites and put them through a selection procedure. And this we did in a collaboration with our colleagues at Baylor College of Medicine, Lorenzo Santarelli in our lab and Chris Thompson, who was a postdoc in our lab, in their lab. Sorry, I left out the P in his name. They took a library of these mutants and allowed them to grow up as fruiting bodies on a single plate, collected the spores and transferred them to another plate and allowed them to grow up as spores. And I think you can see this where this is doing. They did this over multiple rounds, over 20 rounds. So there were 20 opportunities for mutants that had a cheating phenotype to increase its population. And so we could look at the populations after this. So a reminder, I've got the cheater A slide up here again, the cheater that Rich Kesson found years ago. We used that as a control. Remember, it goes mostly to spores. And when we tested the cheater A, indeed, it goes mostly to spores. We also had a negative control, dim A, we knew was a victim. It would go mostly to stalk, and it did that in tests against our strain. So we've got our controls. Then what happened in our experiment? What we did was to take our evolved lines at different generations at transfer 1, at transfer 10, and at transfer 20, and tested them back against the ancestral strain and see what is their representation in the spores. Well, you take generation 1 and test it against generation 0, the ancestor, and it gets about 50% of the spores. Not surprising, there's only been one selection episode and not much opportunity for change. But if you take either population at transfer 10 or the population at transfer 20 and test them back against, in cheating experiments, against the ancestor, you can see that they both cheat. They get 60 or 65% of the spores. So cheating did increase in these experiments. And then, because of this REMI procedure and the insertion into the genome, you can track where those insertions went. And we found that we had at least 160 different REMI insertions that had increased in this study and were therefore potential cheaters. We tested a bunch of them, not all 160. And yes, the majority of them were cheaters, maybe 70, 75%, if I remember correctly. So we have a large candidate of potential cheater mutations. Some of them in genes and some of them in intergenic regions and regulatory regions. So then we needed a population of clones to look at the evolution of the genes. And we had been doing collections over the years, and we selected 20 of our clones from 2 main locations and several other locations across the country, and did genome resequencing. So we had genomes for 20 of these things, and we could look at the polymorphisms in any of the genes we wanted to. And what we found, we looked at it, this is work led by Elizabeth Ostrowski in our lab, and we looked at a lot of possible measures of both balancing selection and directional selection. And what we found, somewhat surprisingly to us, is primarily signatures of balancing selection. Here's one of them here, mean FST. So we had different geographical areas. We could measure the geographic differentiation via FST between those areas for any given gene. And the prediction is if you have balancing selection that maintains 2 genes in stable frequencies, then even when they get separated into different areas and gene frequencies start diverging in those areas, those particular genes won't diverge because they're being maintained by selection at their frequencies. And that's what we found, is that FST, if you take random sets of genes, showed this distribution, roughly an average of about 0.05 in the population. But if you took our candidate sets of cheater genes, or the regions around our cheater insertions, 10 kb regions around them, they showed a much lower FST, as if they were under balancing selection. And we also saw a couple other indicators of balancing selection, lower recombination in those areas and fewer haplotypes. I'm not going to show you those data, but all those data kind of suggest that the stalemate model pertains to a number of these, some fraction of these candidate cheater genes. I should mention that a couple measures didn't line up with that. Tajima's D, which is used to look at balancing selection, didn't show it. Can't explain why some measures show it and some didn't. Now, we followed this up with another study later with a different set of candidate cheater genes that were gathered in a different way and would show perhaps different kinds of behavior. What we did here, our reasoning here was that genes that were turned on or possibly turned off in chimeras are mixtures, genes that are turned on in chimeras, why would you do that? Why would you turn on a gene in chimeras that doesn't get turned on in clonal development? Well, because you're with somebody else. Maybe you're trying to cheat them or you're trying to keep them from cheating you. So what we did was to grow up 2 different clones. Actually, we replicated over many clones, but in a given pair, we would grow up 2 clones to the tight aggregate stage, which is thrown here, where you've got these aggregations that haven't yet fruited. But this is a stage where we believe cheating might be happening. And we also did it for a mixture of those 2 clones together. We got tight aggregates for those. Then we collected them and did RNA-seq on them to determine which genes were being expressed. You're just sequencing the messenger RNA and quantifying that to see which genes are being expressed and looking for genes which are being either upregulated or downregulated in the chimeras. This is work that was done by Sujin No. The graph here just shows one of these diagrams that you get out of RNA-seq experiments, not showing all the genes, but showing the subset of them. Genes are shown on the left axis, and the either clones or chimeric mixtures are shown on the y-axis. And the genes which are overexpressed in chimeras are shown on the top here. They've got more red in the area in the upper right here. And the genes that are underexpressed in chimeras are shown in the bottom here with more expression in the single clones than in the chimeras. So we've got potential cheating genes here, and we can look for our signatures, our molecular evolution signatures of selection in the same way we did before. So again, we looked at a bunch of measures, and this time it came out differently. We did not get measures of balancing selection. Incidentally, this is a different set of genes than the 160 we had before. They could have overlapped, they could have been the same, but this is a different set of potential cheating genes. What they showed instead was a strong signature of directional selection, of the kind of arms race or escalating arms race kind of evolution. The measure we use, the primary measure we use, the best one, I think, is called alpha, the rate of adaptive evolution. It's a statistic which combines the data that you use in a McDonald-Kreitman test, if some of you are familiar with that. Non-synonymous and synonymous polymorphisms, and non-synonymous and synonymous differences between species, with the prediction that in escalating arms races, you'd see an acceleration of non-synonymous differences between species in these arms races. And that's what we saw. Genomic background had these values. Our chimera-based genes had higher values of alpha, more adaptive evolution. And that was particularly true for the genes that were upregulated, which are the ones where I'd expect most. You're turning on genes in order to do your combat. So curious thing, we got one kind of selection measure out of one set of potential cheater genes and another one out of the other. So I'm not quite sure what to make of that. It's interesting. And another thing I have to add is that this result didn't replicate in a parallel study that was done by a different lab. They used a very similar procedure, growing up clones and chimeras, finding genes that were overexpressed or underexpressed in chimeras, but they used different clones, used a different measure of selection, although related to the one that we used. And more importantly, they used different chimera genes that were found at the slug stage, a much later stage. Maybe cheating just isn't going on at that stage, but it's an interesting thing to note. But overall, we were definitely able to find some signatures of the kinds of selection you would expect under evolutionary conflict. Okay, I'm going to finish up with a couple of traits that we think make sense primarily in the context of cheating. That is, if they've had a history of selection for cheating in nature, you might expect the kinds of traits that I'm going to show you here. And I'm going to talk about 2 of them. The first one is a competitive trait in chimeras. And I'm again going to show you a graph from that same study of Neil Buttery's from a while back. So this is from somebody else's lab, but it's been replicated a couple of times. Again, showing just a single clone, but they found this in most of the clones, a single pair of clones, but they found this in most of the pair of the clones they looked at. Again, they were measuring all of one clone and the number of spores it made, all of another clone, the number of spores it made. This is the expected value if you mixed half of each, if something funny wasn't going on. So then, and this is the value of the total number of spores that was actually produced by the chimeras. So here, in the other part of the study, they looked at the proportion of the 2 clones in the chimera. Here, they're just looking at the total spore production. And they found that generally chimeras produce more spores. And that's exactly what you'd expect. If you can somehow sense that you're with another clone, why not produce more spores and take advantage of it making the stalk? And why not make less stalk, because your stalk is going to partly be supporting the other guys and not your own clone. So in general, they're producing more spores. And to me, that really only makes sense in the context of competition. The other trait that's really interesting in this context is kin recognition. Maybe they recognize each other. And in fact, presumably they have to have some sort of recognition in order to do the kind of thing that I showed you on the previous slide. But the kind of recognition we're going to talk about here is kin recognition to avoid being in chimeras. So cells aggregate together and they form these fruiting bodies. A little backstory here. This discovery we first made not in Dictyostelium discoideum, which is a species everybody studies, but in Dictyostelium purpureum, which we were looking at for a few things. But one thing we noticed when we made mixtures of two clones of Dictyostelium purpureum is that they produced roughly twice as many fruiting bodies as a single clone would on its own. And a possible explanation for that is that they're recognizing each other and segregating into separate fruiting bodies. You don't want to aggregate with the other guy. So we tested that first in Dictyostelium purpureum. And we used the standard kind of experiment where one clone was dyed and the other one wasn't, and we mix them together and see how many spores are of that type. But we had a control, that on the left here shows our regular experiment with two clones, one of them died. We had a control for each clone one by itself and clone one died, that ought to produce 50-50 mixtures of spores because it's the same clone. And they ought not to segregate is the main question here. And here's what we saw in terms of the percentage of glowing spores, which would be the proportion of one of the clones in the mixture in chimeric mixtures and the controls. In chimeric mixtures, it was very spread out. You had some fruiting bodies, we tested individual fruiting bodies, some fruiting bodies that consisted almost entirely of one clone, some that consisted almost entirely of another clone, and then spread in between where there was some mixture. And in the controls, they were almost all near 50% where you'd expect. So when you put different clones together, they tend to segregate out from each other. They try to avoid each other. When we looked in discoideum, this is going on to a certain extent too, although the reason we didn't pick it up in the first place in discoideum is it isn't as strong. It seems not to be as strong, and they don't produce twice as many fruiting bodies in chimeras. But at any rate, they still have that behavior. And our collaborators, together with our collaborators at Baylor University, Adam Cuspet and not Baylor University, Baylor College of Medicine, Adam Cuspet and Gadi Shulsky and their student Rocio Bentos, we were able to figure out, and when I say we, I mean mainly they, but we helped them out, we're able to figure out what the genes were underlying this. There were a couple of genes known already in discoideum that were cell adhesion genes, Tiger C and Tiger B, they were called something different at that time, that were co-expressed during development at about the right time in development here. The proteins were expressed on the plasma membrane where adhesion genes ought to be expressed. And the sequence, when you looked at it, had multiple immunoglobulin fold-like domains, which could be the kinds of things involved in recognition. And they had a Tiger C knockout, which when you knocked out the Tiger C gene, the cells would aggregate together and then they kind of fall apart. They'd aggregate together and kind of fall apart as if they weren't seeing anybody that they really wanted to aggregate with. And so this turned out to be the genes that are controlling segregation in discoideum. And that was proved more conclusively in a later paper from the Cuspet-Shulsky labs, where they did some genetic engineering, where they took the standard lab strain and moved Tiger B1 and C1 genes from wild clones into the lab strain. So some of them had Tiger B1 and C1, and other ones had Tiger B1X and C1X. They had several different combinations of the Tiger B1 and Tiger C1. And then they did some of these mixed experiments. In this case, both clones were labeled, some green and some red. This is the control in which You've got green labeled ones with tiger B1 and C1 and red labeled ones with tiger B1 and C1. They've got the same tiger genes, they ought to mix. These ones have green and red ones that have different tiger genes and ought not to mix. And so here's what happens when you see them aggregating. Now, they both always come together in all these experiments. They come into the same aggregations, but what you're seeing here is that these guys mix thoroughly, sort of comprehensively here, and they would produce a single slug from that. And these guys form completely different regions of green and red. And if you follow this further, you would frequently see a green slug come out from this and a red slug come out from this. And then crazily, sometimes later, the slugs later fuse. And that may be why we don't see twice as many fruiting bodies in chimeras here. But they do segregate. They have this segregation mechanism here that apparently keeps them separate for a while. So that's the kind of thing that I think only makes sense in the context where you've had historical selection to be wary of the other clone and try to avoid it to a certain extent. And a kind of clincher in that this is truly adaptive and not just accidental effective adhesion genes is that these genes, the tiger B1 and tiger C1, are extremely variable. If you're going to discriminate against a lot of other genotypes and avoid a lot of other genotypes in the population, you've got to have a bunch of different genotypes at these discriminating genes. And what we see here is nucleotide diversity in all the genes across the Dictyostelium genome. Almost all of them are down here in a low tail, but a few of them are up very high, and they're all tiger genes. Tiger B1 and tiger C1 are the ones we think are important here, but there are other members of the family too that are also variable. We're not sure what's going on there, but these ones are extremely variable, evidently maintained by strong degrees of balancing selection to be able to recognize the other guys. Okay, running out of time, but this is a summary slide. Is cheating an adaptation in the wild? We think, yes, it is. And we have 4 indirect lines of evidence. Do they have a population structure suitable for cheating? Yes, there are some chimeras in nature, enough to make cheating a possibility. Is cheating lost when selection is removed for it? Yes, and that's consistent with cheating being a fitness component in nature. Do you get molecular signatures of conflict in candidate cheating genes? Yes, we do. In some cases, we get signatures of arms races, and in some cases, we get signatures of balancing selection. Interesting that we should get both of those. And finally, do we have traits that make sense primarily as adaptations to cheating in nature? And yes, we have at least a couple of those. Increased spore production when you put 2 clones together, trying to avoid the stalk, and kin recognition to avoid chimeras. So that's my talk today. I'd like to thank John Strassman and all the lab members involved in these projects I've named here, collaborators on these projects I named over on the right. Our current lab members, most of them don't show up in the list on the left because we're currently actually working on some different things, especially on amoeba bacterial interactions. So there's a picture of our current lab group working on these things. And down below, note we've got a postdoc available if any of you folks think you've got something to contribute in this area. We're particularly looking for people who are interested in Dicty as a predator of bacteria. And so check out our website on that. So plenty of time for questions. Thank you.
Thank you, David, very much. Fascinating talk. And now we can move on to our fine session of questions and answers. For those of you who are not familiar with how we run this series, let me remind you that the request is that you simply enter a question mark in the chatting window. If you're new to the series, I would like to ask that you please indicate with an S, that will take S to stand for postdoc or student, because it's a tradition in the series that we're going to give priority to our younger scholars to pose questions. So if you're a senior colleague and you see that I'm not calling on you, it's because some student takes priority over you. So just type a question mark and I'll be calling the questions as they come. Before you go, in case some of you we know that you need to go to something else, let me remind you one more time that next week we're having Aubrey Kelly from Emory University. Let me quickly, I think it's worth it so that you can mark your calendars. So she'll be talking next week about neural mechanisms underlying grouping behavior in spiny mice. That's Aubrey Kelly. Facundo, thanks for letting me know that we have a misspelling. Sorry, Aubrey, we'll fix it for next week. It's Kelly without the U, just the Y, from Emory University next week. But now we want to focus on grilling David with questions. So I will start taking them as they come. And give me a second. It is that, oh, and please don't forget to state your affiliation and your full name. The first question comes from Emmy Slecht. Emmy?
Hello, my name is Emmy, and I'm from the Max Planck Institute for Biological Intelligence, south of Munich. A very, very fascinating talk. Thanks a lot. It's very, very interesting. I have a question about the origin of this phenotype. That how could you explain the evolutionary origin of these stalk cells? It seems like it would automatically induce mechanisms similar to those you would observe in meiotic drive. So what would motivate a cell to take the evolutionary role of becoming a stalk cell? You said that DNA degenerates. It seems like they sort of voluntarily opt for being removed from the evolutionary, from their evolutionary role. That's my question.
Yeah, great question, and it's one that I usually spend quite a bit of time on in these talks. And my focus today was in a different direction, but I should have said this. The answer is kin selection, we think, because when you look at the structure of these fruiting bodies in the wild, they are frequently clonal, usually clonal, and when they're not clonal, they usually consist of 2 clones, at most 3. The relatedness is very high. So if you're a cell that's giving up your life, but helping other cells that have your same genotype, that can pay off in a kin selection sense. And of course, we haven't been able to calculate the costs and the benefits for this because we don't know, how far do they go in nature and when do they germinate and where do they go? We don't know. But it's certainly highly plausible that getting these spores in a slightly better position for dispersal so they can get away from this place where there's no food. And if those spores have your same genotype, that can have a big payoff. So essentially the same reason that our liver cells or our kidney cells sacrifice themselves in order to help the cells that do go on to reproduce.
Thank you, Emmy. Next question comes from Karsten.
Yes, hello, I'm Carsten Schradin at the Succulent Karoo Research Station in South Africa at the moment, where we study small mammal sociobiology. And it's spring here in the southern part of the world, and these birds are breeding like their fitness depends on it. I hope they're not too loud. Thanks for a great and fascinating talk. I have a question to one of your very first experiments where you mixed the 2 clones, and then one clone produces much more spores, and which we interpret as cheating. And you excluded several alternative explanations, including that one clone might make more small spores and the other one more big spores. Now, just from my understanding, did you also check for the same for the amoeba themselves? I mean, these are still all single cells, and it could be that one clone consists of many small amoeba and the other one of less but bigger amoeba, and then it's just the bigger ones are more competitive and thus automatically are the ones that are going up to build a fruiting body instead of the tree. Could you check for this, not at the spore level, but at the individual cell levels that form these fruiting bodies, that there's no difference?
I don't think that's been looked at yet. Let me first clarify. I didn't exclude those 3 explanations for complications of cheating in the lab. I excluded one of them. The other 2 involve trade-offs that very well may be there. And I don't want to exclude them. And that's one of the reasons I wanted to go on and see if cheating adaptations were important in nature. But I think you make a good point. We do know from several studies, including some of ours, that cells that are better fed, in the sense you feed them more glucose, they're going to be more likely to go to spores. Whether that's exactly a cheating adaptation, I'm not sure, because cells that are better fed produce more spores on their own too than spores that are poorly fed. So when you mix them together, they get more of the spores. So yeah, you would expect cells to take, given that there's some competition, that there's sometimes in chimeras, you would expect cells to take advantage of whatever leverage they have. And it could be cell size, that the bigger ones are better at getting into spores, but that hasn't been looked at. Cell size is a little awkward to measure in amoebas because they're doing all their thing, but you can put them in a solution that rounds them up. You could measure them.
Okay, thank you. Yeah, for me, it's a miracle how you can measure anything in them. So, but I can imagine something.
Another interesting thing I can throw in here that's relevant, and it kind of goes in the other direction a little bit, is that another advantage that you can have is being the first cells to starve. So you could take two populations of the same clone and starve one of them a little bit early and one a little bit late, and mix them together. And the ones that starve early are the ones that get into the spores. So we interpret that as these are the ones that have gone farther and faster in turning on their competition genes. We don't know if that's true or not, but that's a possible explanation for that.
Okay, thank you.
I come up next, but before I ask my question, I definitely want to encourage you, if you're in the audience, young student, scholar, postdoc, we know we've been there, but we're always welcome your questions, and asking the first one is the toughest, but then it'll come easier. So sooner or later, you have to ask for the first time. I really, really invite you to do it now. Why wait? There you go, Julianne. Thanks for following. Julianne, where are you? We'll skip myself and go to Julianne. If you can please introduce yourself, name and affiliation, and go for it.
Hello, my name is Julianne. I'm at the Instituto de Neuroetología in Mexico, and working with spider monkeys, so opposite direction, but I guess all of us are. So thank you for your talk. I was actually wondering if you saw also the opposite extreme, like the opposite of cheating. Did you see any association where both of them were better off, and so cheating would actually have been detrimental to the conglomeration of both of them? Or is that something that you don't really see?
Both of them being better off when they're in mixtures? Well,
basically.
I don't think so. I mean, it did show you the result where if they're in mixtures, they produce more spores, but that would seem like it's beneficial, right? Because spores are the things that reproduce. But the corollary of that is that they got a shorter stalk. And so my thinking is that they're probably going to do worse in a natural situation. Well, okay, here's a circumstance. I think there is a benefit. When you aggregate together, you make a larger slug. Larger slugs can migrate farther. And so if you're down under the soil, under the leaves, and you want to get to the surface where an insect's going to grab your spores and take them somewhere, it's better to be in a big slug. And so there's an advantage for joining together. So they're pretty complex dynamics that, as I showed in those last few slides, pretty complex dynamics where, you know, they do come in together. They form separate areas. Two different clones form separate areas of that aggregation, and then they may shoot off into different slugs, and then the slugs may recombine to make a larger slug. There are complex things going on, and I think your thought is good, and that being together can have the advantage of getting you better dispersal in the slug stage.
But there are no Dictyostelium that are like constantly together that actually prefer to be together? No, you haven't seen anything like that.
Not that I know, but not very many species have been looked at in this regard. Here's a curious thing. Your questions keep triggering things that I didn't think I was going to talk about. So I showed you the Dictyostelium purpureum, which segregates pretty well into different fruiting bodies. Dictyostelium discoideum, they segregate into different areas of the slug and sometimes into different slugs, sometimes not. We've actually done some studies where you mix Dictyostelium discoideum with Dictyostelium purpureum, two very distantly related clones, different species, and they do mix to some degree. It's very curious. And I don't know if that's accidental, that they don't normally encounter each other in nature, but maybe they're mixing on purpose in order to get a larger migratory stage.
Mm-hmm.
Although,
Mm-hmm.
adding one thing, just a bit of biology, Dictyostelium discoideum is unusual among the, there might be 200 species of Dictyosteliums in producing a slug, a migratory slug. Very few species do that. Dictyostelium purpureum and the others, instead of producing a slug, they start producing the stalk from the very beginning. And the stalk will run along the ground or it'll run up through the soil. It's kind of doing the same. The stalk has much more variable length. It keeps growing until they decide now's a good time to produce spores. So when you mix the two species, you get some sort of hybrid behavior of, sort of stalk, sort of slug. But they do combine. That's strange.
Interesting. Thank
Thanks.
you.
Thank you, Julian. There you go. So we're going to skip Adriana and we're going to go to Sylvia. You're next.
Yeah, hello. Sylvia Kramer from the Institute of Science and Technology in Austria. Hi, Dave. Thanks for the very nice talk. I was just wondering, coming back to the very last point, you mentioned that these, particularly in Dictyostelium purpureum, there is avoidance of coming together of the different clones. Do you think that both partners are or should be equally good in avoiding the other, or would it maybe be even beneficial for real good cheaters to actually not avoid the other one, but only those ones who would not be good cheaters to avoid?
Yeah, that's a good question, and not one I really have an answer to. It's something I have thought about. If you're a great cheater, why not combine with somebody else? Mm-hmm. And maybe there's some of that dynamics going on in there, but we don't know. How could we tell if one clone was trying to join and the other one was trying to lead? Maybe there are ways.
I thought about, I've been working in my diploma thesis many years ago. I was thinking about speciation, and there it was also so that like one species was always a bit more choosy than the other one, whereas the other one didn't care, like in female choices. So I was just thinking maybe there's something related to that also in Dictyostelium.
Yeah, and maybe the answer lies in this phenomenon that when you get mixtures together, they produce more total spores. Mm-hmm. So you can be a better cheater and you get a higher proportion of those spores, but actually you're not getting enough stock because the other guy's producing more spores as well. But I don't discount the possibility of what you're talking about. It's an interesting idea.
So these many spores are actually coming at the expense of the stalk length. Is that what you're saying?
Yes. Mm-hmm. Typically, we don't measure stalk lengths because it's a hard thing to do, but in a few studies, it's been done. And yes, you produce more spores, shorter stalk.
Okay, yeah.
Sylvia, are you a professor or researcher at the institute or postdoc? What are you doing? Mention your diploma thesis.
Oh yeah, well, the diploma thesis was ages ago. No, I'm a professor at the IST. Yeah, working on social immunity in ants actually. So working on social insects and how they cooperate for disease defenses.
Great, thanks.
Yep.
Shakti, you're next. See, they just needed some prodding. Next time, you cannot wait for next Tuesday to start asking questions right away. Shakti, you go.
Thanks, David. That was an interesting talk. I guess The prodding is that the line of questioning is kind of coming from what's been said before, so that's kind of triggered.
Can you please introduce yourself? You can let us
yeah,
know
my name
where
is
you're calling
Shakti
from.
Lamba. Until recently, I was a senior lecturer at the University of Exeter, the biology department, behavior in Cornwall. But at the moment, I'm looking after my child. Yeah, so I guess my question is also related to that last experiment where you have within the same spore, the two different types aggregating at either end. So obviously they're showing some tendency to be with like type. On the other hand, they are also together in one spore. So I'm wondering whether, but you had two examples. One was where they were all mixed up together. And then there was one where you had these two different halves. And why was there that difference? Is that to do with two different genes or is or
You mean those two images on the same slide?
Yeah, on the same slide.
Yeah.
In one you had the spore, they were all mixed up, and in one they were two different halves. So why?
Yeah, so on the one where they were all mixed together was where it was essentially the same clone. They both had the tiger B and the tiger C alleles, but one produced green fluorescent protein and the other produced red fluorescent protein. So it was simply a control. When you have the same clone coming together, do they mix thoroughly? And the other
one had two different
tiger proteins, so that when they came together, the hypothesis would be they would adhere only to their own type and form distinct regions.
Yeah, but on the other hand, they do, like you said, also adhere to the other type, and sometimes they move away and then form bodies and do adhere. So I guess my question is about the boundary where they adhere. Because you've got the like types adhering to each other, that's kind of in a way understandable. But then you've got this boundary of interaction where two different types are having to adhere to each other in order to be together. And I'm wondering what's happening over there essentially, and whether you could investigate that boundary in terms of changing the shape of it. So again, like if you ran a selection experiment, and here I'm now just kind of, ideating, I don't know if it's possible, but for instance, if you change the shape of the boundary so that it's longer, and they in order to form one whole, they have to, you have to have a longer boundary of junction or a shorter one. So that would obviously change the selection pressure in terms of what is required to be together as a whole. Am I making sense to you? You know what I'm saying?
Yeah, at least part. I'm not sure we can do that. I'm not sure how we would change the boundary. But yeah, that's interesting. What I think is probably going on, so I'm not a physicist, but I think what's probably going, they're attracted to cyclic AMP. That's true for all clones. So clones are always going to be attracted to the strongest site of cyclic AMP signaling, and they're going to end up in the same place, and that's what they do. But if you have two equally common types that differ in their adhesion, they adhere more to their own type and less to the other type, and they're attracted to a common place, I think that's the shape you're going to end up with. They're all in the same place, but you get a dividing line between them.
I guess my question is that if they have this tendency to aggregate with like type, then which are these? But there are certainly some cells which are agreeable to form that boundary of juncture with another type. So something different is happening. So if I'm a cell which is kind of further out in that spore, I'm surrounded by my own family or whatever. But then if I'm in on the boundary, so I'm actually a human ecologist. So I would say similar things happen with humans at migration boundaries. You have different tribes and communities living in different areas. And of course, you have villages distributed all around, and you have those that are more secluded, which might have a lot more endogeny, and also, their people are interacting only amongst their own. But then you have the boundaries where something different happens because and at the boundaries, people have to be, for instance, one repercussion is that you might speak multiple languages, because you are at that boundary. So I'm just wondering in terms of also relating to the cheating and cooperation. What's happening with those cells that are kind of willing to in case that you're forced into it, you don't have a choice,
I don't know, but that would be interesting if you could somehow isolate the cells at the boundary and see if they're doing something different.
Yeah.
So I'm following, if I may, because I find it fascinating. This is what I enjoy of this hour, and we're moving back and forth now between humans and amoebas. So are you asking if I'm following you well, because we were literally discussing this with one of the students in the course that we're doing, where they get to watch the videos. She's not here today, but I'll ask her to watch this discussion we're having. Shakti, are you asking whether there may be a distribution of a certain trait that we don't know what it is, that may change, and you may end up, I study non-human primates, but I can relate to what you're saying about humans. Suddenly, there is some personality type that makes these people, but not the others, be more daring, walk further, and therefore they end up in boundaries. That there is something that it's not a random distribution of those traits that may then, they may influence or they be associated with where things happen or not at the boundary. Did I follow you well? Is that?
Yeah, that and also what the repercussions of that might be, because if you have to have, that allows other possibilities. Because if you don't have those types, so in I guess what I'm saying is that then therefore there might also be selection maintained for those types, because without those types you can't meet another type. Yeah. If everybody has the phenotype that they only want to be with like, which is usually what's required in any case for cooperation to arise in any system, you need that kind of like-to-like association to occur, that mechanism that allows you to assort with like type, which is what we're seeing here as well. But so that's fine, but then that doesn't allow a certain other kind of and other levels, or you can think of it like that, or other types of cooperation to occur, because it doesn't allow, if everyone's always going away from the opposite and going towards like, so you have, so maybe something that allows maybe some individuals or some selection of the set of the population which is more amenable to meeting unlike type, while still retaining its affinity to like type, I suppose in some ways it's like, cities are described as melting pots versus salad bowls. Like a melting pot is where all the cultures get combined and you get a new flavor. But then a salad bowl is where the salad is good, as a whole it tastes good, but then everything also retains its own character, So I guess I'm in a way the graph on the right hand side, the picture on the right hand side where you have those two things, that's like a salad bowl, whereas the other one was more like a melting pot. So I'm wondering whether, but to maintain the salad bowl also you need some adhesion, you need that oil or the whatever that makes it a salad,
Yeah.
So I'm wondering about that basically. I'm just
in this salad, the cyclic AMP may play that role, because it's just causing everybody to come together. There may or may not be some adhesion between the two halves of that. They may just be sticking. I think there probably is. I think there's probably some binding between different tiger genes, but just weaker. But we don't really know that yet.
Thank you.
Interesting set of thoughts.
I'm going to now ask a question myself, because I think that these are very basic questions that maybe help others picture what's going on. Can you give us some quick answers to the natural history of amoeba? So I was writing that down. How big is an amoeba? When we're looking at those videos or photos you showed us of aggregations, I always then I realized that even when you tell me the size, this is like discussing cosmology or astronomy, they may tell me 6,000 light years, and I still cannot really internalize or comprehend. But can you give me a little bit of a sense of the size when we're talking about how far they can disperse, right? The aggregates, or what is the unit of measurement? If you can measure, what are we looking at?
Okay, so amoebas are about 10 microns, if that helps.
Yeah, well, no, 10 microns.
Yeah, so they're much bigger than bacteria. They make sense. They're consuming bacteria. We think they have to eat roughly 1,000 bacteria to gain in size enough to double. Now, when they come together, the aggregations can be different sizes depending upon how many amoebas there are coming together. But large ones are macroscopic. You can see them. So we could go out and look at deer feces in the wild and see there's a fruiting body. You might miss a very small one, but it could be a millimeter tall, sometimes taller in the lab. The slugs are also visible, and slugs might migrate a couple centimeters.
Couple centimeters.
Yeah.
Okay.
So
I
really
mean, I like
go longer. Yeah.
Yeah, good. Yeah. And then I don't remember who asked this question, but got me thinking. You were telling us before of the limitation, the challenges of really studying the amoebas in the wild, but you've been talking, and of course, there's a taxonomy of amoebas. It goes down to species. What are the criteria when you are classifying taxonomically amoebas for deciding on species? I mean, what are the, we know that each discipline kind of works paradigmatically within its own boundaries to decide what we're going to accept. It's a different species or not to some extent.
Yeah.
But so how do amoebas get classified in different species?
Yeah, I mean, I'm not a systemist, and I'm not particularly good at that, but it's primarily, historically, it's been morphological, and the amoebas of different species look pretty similar. I don't think they're used very much in the classification. So they're using the social stage. The patterns of the aggregation streams as they come together can be different. The shapes of the fruiting bodies. Some species have branching fruiting bodies instead of the straight ones that we saw. The size, some are very big, some are very small. A little bit of color variation. I mean, basically things that you can see through a microscope. Of course, people are now starting to sequence things and look at the molecular, and it's turning out to be true that usually when you look at a well-distributed species, a common species, it may contain more than one species in there. That was true of Purpurium when we looked at it. That's at least 2 species. Dictyostelium discoideum itself, it's got an interesting range, by the way. It occurs in Eastern North America. We're right about at the edge of its range here in Missouri. And it also occurs in Eastern Asia, in Japan, China. And why that distribution, I'm not sure. But there are also, if you go down south from here into Central America, you get some discoideum-like things that have sometimes been called that. But we think most of them are one or more different species based on their sequences. So the taxonomy is not, the short answer is the taxonomy is not in great shape.
Thank you. So now, Adriana, thanks for your patience. We're going to go to Adriana, and after Adriana, it's going to be Clara.
Hi, David. Thanks for the super nice talk. I'm Adriana Maldonado. I'm professor at the Universidad de Rosario in Colombia. And I have 2 questions, and I think they are from like the opposite sides of the story. So the first one goes when the aggregation is happening. Have you ever tested or you know about sort of associative aggregation? Like some cells that are alike tend to aggregate more than others, or is just a spatial result that you see in this aggregation when they go from just single cells into slug? I don't know if I'm making sense of what I'm asking.
I'm not sure. Are you asking whether some cells prefer to sort and others don't? Or, yeah. Well, there's this loner phenomenon where certain cells do not get into the aggregation. And we're not entirely clear yet whether that's something they do on purpose, because if you leave some cells behind, they'll be able to reproduce quickly, faster than the spores would, because spores are encased and they have to germinate slowly. Or they might just be accidental. They're cells that were attracted to the cyclic AMP and trying to get into the center of this aggregation just didn't quite make it. I don't know.
Yeah, I was just thinking, okay, well, if like, if you have 100 cells around here and then suddenly you have this harsh environment and they have to start aggregating, then who gets to aggregate and who doesn't, and why? What are the rules to get into the aggregation, right?
Yeah. Well, there's certainly people who've worked, it's not a literature I follow closely, but people who've worked on this sort of cyclic AMP dynamics. So what happens is cells start secreting cyclic AMP and also sensing it and moving towards the strongest direction in which they sense it. And as they move, they'll be receiving the cyclic AMP from one direction and then secreting it back the other direction. So as they're moving, they're tending to attract other cells to move in the same direction. And so I think there are chance centers at the very beginning where the signal is strong. Maybe you happen to have 2 or 3 cells right next to each other who are all secreting. And so therefore they produce a stronger initial signal than other localities. And that will then be the nucleus of a larger group where the signaling gets stronger and stronger as the group size grows. But pretty much everybody is moving towards those aggregations or trying to.
Interesting. And so the other part of the question goes on the other side. So you have the stalk, and then I'm assuming, or I don't know, because I don't know too much about the natural history, but they will vary in size, right? Will the taller ones have more fitness advantage than the smaller ones? And I would assume because it's spores, then you can send them further away if you're taller than everyone else.
Right.
So there might be also a competence with other stalks to be larger than anyone else. But then you mentioned as well that the slug somehow has an influence on this, the size of the slug. So that means that it's sort of predetermined by the size of the slug, or can they grow somehow after they start becoming?
So there's not much when they're starving, the signal for aggregation is starving. So they're not in much position to be growing and dividing. There probably is some small amount of cell division at the first. The cells that are close to cell division may go ahead and divide at the beginning of the aggregation period. But there's not much opportunity for growth during this process. It's a process of division of labor and sorting out of the cells into different categories that are going to perform different tasks, but without much growing. I don't know if that answered your question or not.
Yeah, but then how that translated to when you already have the fruiting body? Do they?
Oh, doesn't matter.
The size of the fruiting body, does it?
We think it does, or at least if the size of the stalk doesn't matter, then why bother to produce a stalk at all, in some sense. But it's hard. That's a hard thing to manipulate. We don't have good ways of manipulating stalk size, with one exception. We did an experiment where it's sort of a funny experiment. We used Drosophila as a model insect vector. It's presumably not the real vector out there in nature, but we had it in the lab, so we used it. And so we had some plates where we allowed fruiting bodies to form normally, and other plates where we allowed them to form normally, and then you kind of bang the plate a little bit against the table, and that causes all the spores to fall down to the bottom. It causes them to collapse. The spores are still there, but they're not on top of stalks. And then we let Drosophila loose in there and recorded how much they picked up spores. And they did pick up more from the plates with the intact fruiting bodies and not from the plates where the spores had fallen. So that shows having a stalk is important, at least with Drosophila. But it doesn't show that different gradations are important. That would I wish I could figure out a way to do that.
That would be great.
Yeah.
Thank you.
I think that Clara, you're next.
Okay, Clara B. Jones, retired, social evolution. First of all, I want to say that I'm honored to hear you talk about your research. Secondly, that 2017 paper comparing Fisher and Price was just amazing. So thank you for that. And since we're near the end of the questions, I'd like to ask you a pretty general one. That is, in 2022 and maybe elsewhere, you said that Hamilton's rule, quote unquote, kin selection, explains most or much of social evolution, but not all of it. Can you say a little bit about what you think Hamilton's rule does not explain and why you think it may not be a quote unquote general law?
Oh boy, great question. Discuss that for a long time, but I'll take a little whack at it.
We should have a seminar on that, Clara. We should just invite him to discuss kin selection theory.
Yeah, so it partly depends upon, this is a question I was asked earlier, what your definition of social is. And the definitions could be very narrow or very broad. A broader, a very broad.
Could I interrupt?
Sure.
I'm defining social the way Hamilton does in 1964, which is limited to cooperation and altruism.
Okay, but I'm not sure if you're quoting me correctly. So I would think that kin selection, Hamilton's rule in some form would explain most of what we see in social evolution. There are controversies about how you should view things when fitness costs and benefits don't add neatly together. That is when, you do an altruistic behavior, nothing happens. Your partner does an altruistic behavior, nothing happens. But if you both do one together, something happens. When there are these non-additivities in the situation, you can derive versions of Hamilton's rule, which seem to be correct, but the costs and the benefits and the relatedness don't quite mean the same thing. There are complexities in this. And this is what underlay a good bit of the controversy from that Nowak and Wilson paper on eusociality way back in 2011. Wilson, for some reason, seemed no longer to believe in kin selection, but in fact, kin selection was in their models. I think Nowak and Tarnita, the other author, I think they believed in kin selection, but they didn't think inclusive fitness always worked. And that was because of these non-additivities. So it can work if you define the variables in different ways, in which case kin selection explains things nicely, but they didn't want to do that. They didn't want to define those variables in different ways. I do think, the summary is, I think it's an extremely powerful theory to apply and has been substantiated in a lot of studies.
Thanks.
I think that it's already 20-1, and David has several individual meetings with members of our labs. So I think it's fair to him that we give him a break, a little bit of time to recover and prepare for his meetings. I want to thank you all, very, very especially Julian and Shakti, because correct me if I'm wrong, but I think it's the first time that you pose questions. Maybe you've joined the meetings before. I hope to see you again. Clara, of course, you're always joining us and contributing with your ideas and your questions, and I thank you as well. And Karsten and Adrian, I won't thank you. We're all doing the same, so thank you for being here. Before we go, remember, those of you, the YouTube community, we welcome you as well. We thank you for joining us. Next week, Aubrey Kelly from Emory University, discussing neural mechanisms in in mice, in spiny mice, right? So thank you all. And with that, I suggest that I finish